Welcome to Careers in Discovery, where you'll meet scientists who've forged outstanding careers in biotech and hear about what they've learned along the way. This podcast is brought to you by Singular, building brilliant biotechs. Orla Cunningham is the Chief Scientific Officer of Granular Therapeutics and Ultrahuman, companies developing novel single antibody therapies for a range of diseases. Orla talked to us about her fascinating career to date, as well as the therapeutic potential of mast cells, how to get the best from your CRO partners, and why communication is the most important skill scientists must develop. This week on Careers in Discovery, I'm really pleased to be joined by Orla Cunningham. Orla, welcome to the show.
Thanks, Tom. Nice to be here.
Fantastic to see you. So Orla, we always start by talking a little bit about what you're up to now, and you're involved in multiple things, somewhat related. Tell us a little bit about Granular Therapeutics and Ultrahuman and how that all works for you, and give us the sort of overview of what you're up to at the minute.
Sure. So I actually started with both companies around the same time. I actually left a position in Pfizer during the pandemic back in December, 2.5 years ago now. And it was really through conversations with an ex-colleague of mine, Johnny Finley, who I believe has actually been on the show himself. Yeah. We were very close colleagues across many years, probably about 10 or 11 years in Pfizer. And he left the company before me to establish a series of antibody therapeutic companies, all VC funded and initially set up to be single asset. So I was always very much in touch with what he was doing and excited at some of the progress he was making. I mean, initially he set up tons of companies all based around single antibodies, and, you know, some fell by the wayside, some were becoming quite successful and getting quite busy and difficult for a couple of people on their own to manage. So yes. I stayed in touch, and I was really excited at the opportunity, I suppose, to be much more involved in every aspect of therapeutic development. I think from my role in Pfizer, I was leading an antibody discovery and technology team, and we were touching across all therapeutic areas within Pfizer, but with the unique, I suppose, and very specific role of antibody engineering. So I always felt we were creating these fabulous assets, but they were kind of moving on to different teams within the organization, and very much losing touch, moving back to the next program and the next project, and not really being part of the development of those molecules and really driving the efforts to bring them to patients, and having the opportunity, I suppose, to be a champion for those individual programs as they move forward. So with Granular and Ultrahuman, these are two, again, single asset companies. Granular is very much focused on the development of next generation therapeutics for mast cell targeting, particularly. We're very interested in the sort of pleiotropic role of mast cells as sort of master immune cells within any different chronic inflammatory and allergic conditions. So there we started with one antibody with the very specific aim of generating a bispecific molecule that could selectively modulate mast cell function and steer clear of other, hematopoietic stem cells, for example, and other immune cells that we really don't want to be targeting. As the programs have developed, I suppose we now have actually two very exciting molecules moving forward. With Ultrahuman, it's very different. It's immune oncology. It's a very, very interesting story where the kind of lead asset came from originally, and sort of developed, I suppose, from this idea, I suppose, that the tenet of monoclonal antibodies being uniquely specific for target. And one of the things that myself and Johnny had been working on while we were at Pfizer was this idea that actually therapeutic antibodies are not, uniquely specific. And I think that idea is very well known in the area of diagnostic antibodies, where for many years, people that sell antibodies have been doing a lot of QC validation and showing that actually a lot of these antibodies are binding to more than one target. So it turned out that this antibody the Ultrahuman 8 lead antibody came out of this observation. It's an anti-PD-1 antibody originally that is being used to dose patients, globally and very successfully. But because there are so many PD-1 molecules in the clinic now, we know that there's a very specific adverse event profile associated with PD-1 targeting. And what this antibody was showing up is actually a secondary phenotype. So a lot of dose patients were turning up with this spontaneous hemangioma, which we thought was very interesting. And Johnny and the guys went on to show that this was actually related to an off-target event and a clinically relevant off-target secondary target. It turned out to be binding to the VEGF receptor 2, and not only binding, but actually agonizing that receptor. So agonizing angiogenesis, if you like, which is not what we really want to do in the context of tumor growth. And so the first step was clever antibody engineering to return it to be a PD-1 specific antibody. But then, of course, a lot of the really nice combination therapies that have been going on, combination trials in the clinic right now, have been showing that actually one of the few synergistic combinations turns out to be blocking PD-1, PD-L1 signaling at the same time as blocking angiogenic pathways. And combinations from Roche, with Tecentriq and bevacizumab are actually showing really, really nice combinatorial efficacy. And they published a really nice paper last year that really went into, the sort of biomarkers associated with that synergy. Right. So we thought, well, actually, this is a really nice combination. Could we go back and again apply those antibody engineering principles and turn this this dual specific antibody into a beneficial thing? And we actually flipped that VEGFR2 agonism into an antagonist. So now we have this really nice, and it's not even a bispecific, it's a single antibody. So none of this complex, manufacturing that's associated with the generation of classical bispecific antibodies. It's a single antibody and it antagonizes both of those pathways in what we believe is a very synergistic way. So that's, my role, I suppose, and it's the same across both companies. On paper, it's a 50-50 split, but really it's just, generation of all of the preclinical data that we need to move the assets forward. And I guess I didn't mention, but both companies are completely virtual. So we are, this is me, this is where I am, talking from today is my office. And really it's interacting with multiple different contract research organizations, collaborators to achieve all the experimental goals we need. So for Ultrahuman 8, we have a lead molecule. We've done all of our initial CMC assessments. We know it produces and behaves very much like a really nice antibody. We believe it's very, very developable. And we're at the next stage now of moving into a round of fundraising to help us launch that full CMC and GLP talks for the next stages. So for Granular, I would say we are a little bit earlier. We're moving 2 lead assets in parallel. So a lot of work going on there. And initially it was myself and Johnny, and we have a chief operating officer, Jamie Coleman, who's involved in both companies as well as the many other things those 2 guys are doing. With Granular, because we're moving, we have 2 assets. We recently have expanded and brought on a new CEO and a new chief development officer. So we're at a really exciting stage of expansion. We have the funding that we need from our very generous backers at Medici, and we're really moving forward there into formal CMC for our two lead assets. So it's been an absolute whirlwind, I would say, the last 2.5 years. It's completely out of my comfort zone, I would say, for the vast majority of that time.
Okay, that's good to know. And I want to talk a little bit more about some of the science behind it, but before we do that, I suppose in some ways, sitting across these 2 companies, because they're single asset companies, is similar to managing a couple of different projects. But I would imagine there are extra things that come with them being 2 separate companies and extra elements to your role as a result of that. What is it that's different about that? Because I think this virtual model is becoming more viable, right? And so then people can potentially get involved in different things at the same time.
Yeah, completely. I think, my experience, and I guess I've always, I'm not one of these people who really knew from the very beginning what it was I wanted to do. I wasn't even sure that science actually was you know, where I saw myself. So I've been a sort of a generalist, I would say, from the beginning. And I think when I took on the role from academia, I mean, I did a postdoc in cancer cell biology, and then I saw this opportunity to join what was a kind of a nascent research group, originally with Wyeth in Dublin. I was in Italy doing my postdoc at the time, and I thought it was a great opportunity to come back. At the time, I would have probably classified myself as a biochemist by training, with a little bit of cancer cell biology. And this role was very much antibody engineering, and it took me a long time to feel comfortable enough, I suppose, and proficient enough in the technologies to call myself an antibody engineer. I think finally, after, you know, the acquisition by Pfizer and another probably 10 more years working as an antibody engineer, I kind of believed I had become an antibody engineer. I think what's very different in the role I have now is that, you know, I had a very specific technology role, I suppose, and thinking about what is the biology, what is the type of molecule we want to make, and then making that molecule to very specific, you know, I suppose, characteristics, and then handing it on. And I think in these two companies now, you know, I have the role of coordinating all of the activities. I think all of the preclinical in vitro assays that we need, all of the sort of early stage production, manufacturability, thinking about, you know, what are the disease indications we want to go after, what are the relevant in vivo models, thinking about how to start those CMC campaigns. You know, it's really thinking about interfacing with regulatory. I mean, all of these things that you know, I would have had no interaction with or no contact with at all in a large organization. I think it has been, you know, really fascinating. And I think really what has been, I think the nicest thing for me is, you know, being an antibody engineer, I think it's very, there's so many things we can do now. I think making antibodies has become, you know, a really simple thing to do. And so I think there's a big area that we're missing. You know, we've always thought about how do we engineer specificity or affinity, or, you know, and a really, you know, I think we should be migrating much more towards, you know, we can do these things now. So the big thing that's missing, I think, in things like, you know, if we think about Ultrahuman 8 and oncology, you know, thinking about, we can have a cytokine and we can add on a CD3 bispecific T-cell engager, and let's add on a CD28 agonist into the mix. And all of these things, of course, you're adding efficacy and you're adding potency, but you're adding an incredible amount of tox. And that's something that we haven't really got any better at all. And with combination after combination, you know, that's something I think we really need to get better at tackling. So I think the exciting challenge in both of the companies that I'm working for, while there are differences at the stages they're at, I think the similarities they have is we want to do things that other people are already doing, but we're not just thinking about potency. We're thinking about that, you know, that real important balance between potency and toxicity, and between site of action efficacy and systemic safety. And so thinking about how to use all of that antibody engineering expertise, I suppose, to try and equally tackle both of those challenges in parallel.
Yeah, that makes sense. And I suppose thinking about the long-term regulatory requirements, tox requirements, all this kind of stuff from the beginning, I would imagine puts a different spin on how you conduct the early research as well, and how you bring these programs through.
It does. It also presents, I suppose, in itself a number of different challenges, because while, you know, the sort of in vivo models we have at our disposal have really significantly improved, and, you know, everybody's working with really sophisticated humanized mouse models now, I think the challenge that we, of course, have is that because we're trying to look at both site of action, so looking at human tumor, human immune system, we're also trying to think about normal physiological systemic functions. So we're thinking about where VEGF-R2 is most important outside of the tumor, which is the vascular endothelium, and critically important for many physiological functions. But we don't yet have a mouse that has human immune system, human plus human, you know, VEGF biology. So we want to do that. And that's one of the big challenges we have is really thinking about how to translate from our kind of 3D co-culture systems into something a bit more sophisticated. Yeah. I think there we have to start thinking in different ways. Well, PD-1, VEGFR2, these are pathways we understand. There's tons of clinical data that suggests that synergy is there, that it works for patients. So, you know, maybe there's a different way to get there without having to go through the hurdle of not very realistic, potentially not really translational in vivo models. So it's a constant series of challenges, I think, that are really interesting. And I think while, you know, the sort of biology between both companies is very different, I think the sort of challenges we have are kind of similar. And I think what's really critically important, and I think has been the sort of guiding principle for me the whole way along this pathway, has been that there is never going to be a system that will answer the question. Yeah. So we have to start to pick apart what are the different pieces and think about what's the biology or the assay or the model that will help us answer that piece of the story. And of course, critical in bringing all of that together is convincing somebody in the conversation that it's okay to do that, to pull these pieces apart and answer these questions individually and build the story in that way that builds confidence. So in that way, I think another thing that has been really radically different from my time in pharma compared to this is really the mechanism of communication and the way of telling the story is really different. I think, you know, the audience you're trying to convince is really different. So yeah, that's been interesting too.
Yeah, and I suppose you know, there's always lots of audiences that you're trying to convince, right? Whatever context you're in, but they're probably slightly different. And I suppose, you know, one of the things I was interested in was your experience with moving to this virtual model and the differences between it, because I'm sure that you used CROs at Pfizer, and I'm sure that they do extensively as a company, but there's a lot of in-house resource as well, right? And that aspect of it is probably quite interesting in sort of making sure that the work you need done is being carried out in the right way by the CROs. And of course, the quality of CROs has improved massively over the last several years. And so that will help. But also, you know, you've got this fundraising element now to biotech, whereas I suppose you would need sponsorship for projects in a larger company. But it's a different conversation, right? So just interested in your observations around that in terms of communicating, in terms of how this virtual model's worked for you and the differences you've noticed in moving to it.
Yeah, I mean, it's definitely true. I think the difference, I think, within pharma is generally the way most companies operate. I suppose it's a very matrix style where you have the biology experts who think this is a great target, and we think about what is the best way, you know, to challenge that target or to start to explore that target in terms of, you know, what is a therapeutic modality we think that could be of most interest. And therein starts the first sort of restriction, if you like, because there may be IP reasons why, you know, this is our platform and we shall make this platform work irrespective of what the biology question is. Whereas I think in this way, we're completely agnostic. We don't care what the platform is, or it's like, what is the biology and what is the best way to interrogate? And I think that's a really good example of that is what's happened at Granular is, you know, during the course of our explorations of our initial modality, which is as a bispecific antibody, we kind of realized that actually, you know, there's other indications where part of this modality or part of this mechanism could work really, really well. And so we've really, you know, because of the sort of, you know, initial antibodies we were discovering, we realized actually there was ways to branch into different indications with a different type of modality, but using a lot of the lessons that we had learned. And so this kind of flexibility and, you know, this idea of being agnostic to a platform or being restrained by a specific IP, you know, you're able to think in a much more open-minded way, I think, about what target you want to challenge. And again, you're not restricted by the tools that you have in-house. You can think who's out there doing it, who are the people that can provide this service. And again, it's this service, and you can step in and then you can move away when you achieve that goal and move on to the next, you know, very, very quickly flipping between different service providers. But I think, I mean, critically, again, communication is key. You know, if you're working with a CRO, I think it's very easy to say, well, we know exactly what we want to do, and to be prescriptive and say, we have all the knowledge, you don't need the knowledge, you just need to provide the service. But of course, you know, for anybody to have a proper scientific engagement and to best utilize people's skills and technologies, I think communicating the story is really important. And we do that. I think we really try to do that very well from the beginning, that it's a project team. It's not a service, it's a collaboration. And we really want to partner, I think, with all of the CROs we've worked with. I mean, we've been very lucky. I think, you know, Johnny was at this game for a few years before I joined, and already had a great, you know, kind of baseline of providers that we use for routine production. And like you say, I mean, that this quality of scientists that is engaged in these kinds of services now is really, you know, second to none. I think lots of people have come from big pharma, have had excellent training, are very, very experienced. And again, you know, coming out of that environment, want to have the ability to be a little bit more, or a bit less restricted, I suppose, in how they're applying their specific skill set. So, you know, communication, I think, is key in establishing that relationship early on, that it's a collaboration, that, you know, people get so engaged in your project, they become part of the team. And, you know, so really I found it a really, really easy model to work in. You know, I came with, I suppose, certain preconceptions as to how it was going to be. And it certainly, you know, my experience has been massively positive, I would say. You know, because people are really expert in the services they're providing and are really open to having, you know, a really robust discussion and back and forth and collaboration about the steps to take next. So,
and I suppose great. if you're running a biotech company, there's only ever going to be so much demand, for example, to in-house have real deep expertise in, I don't know, rat-based models of disease, right? Whereas in a CRO, because you can provide that to lots of different, you can really invest in developing people's expertise, and they do it in all these different contexts, and they bring that to the table. I think potentially where some companies fall down with using CROs is that they try too hard to protect their IP, which of course you've got to be careful about, but as you say, don't bring them in as part of the team. It's at arm's length. And then of course, you know, without that context, as you talked about earlier, in terms of understanding the toxicity requirements early on, it's just going to be harder for them to do their job, right?
Exactly. And I mean, also for me, I mean, I'm coming in and it's okay if I'm engaging with somebody in terms of antibody discovery or engineering. I feel like I come from a pretty well-informed and experienced position, you know, to think I know what it is we need. But there are many other situations where I'm reading the literature myself. I'm a complete newcomer. I'm coming with ideas, but I absolutely need people, experts with experience, to help me understand, well, actually, I don't think you should be doing this. This would be much more suited. So it's certainly a two-way street. And I think, again, in both elements of that, you know, coming from a big organization where there are a lot of restrictions about what you can talk about and disclose, and you know, it's very hard to have that kind of expansive discussion and brainstorming and really figuring out how best to collaborate, because information is restricted, I think, in both directions. And by being restricted in one direction, of course, you're making somebody else feel like they don't want to really share. You're not creating an open environment where that sharing, it just comes naturally. So I think it's been, from that perspective, it's been a very positive experience.
No, absolutely. And then before we talk a bit more about your career specifically, Orla, I wanted to pick up on, you talked about mast cells earlier, and the importance of them, the role that they seem to play, and the potential therapeutic potential of targeting them. I've said potential too many times. But for those who might not be familiar, can you give us, without giving away the family secrets, obviously, just a introduction to that briefly?
Yeah, I mean, I suppose it would be really nice. We don't have any visuals here, but certainly, you know, mast cell is at the center of, you know, they release huge amounts of chemokines, cytokines, you know, digestive enzymes. It really just an explosion of you know, stimulatory molecules which go on to stimulate downstream any type of inflammatory cell you care to mention, you know, B cells and T cells and all sorts of, you know, really important downstream immune signals. And so, you know, there are pathways that, of course, I mean, there are, and mast cells, the reality is we know very, very little about mast cells. I knew nothing about mast cells before I started. But, traditionally, historically in the literature, there are two main types of, connective tissue type mast cells and mucosal. There's been a really recent paper published by Minicus Gaudenzio's team to suggest there are up to 7 different subtypes of mast cells associated with different organs, and they have very different, potentially very different roles. So, obviously the classical IgE dependent signaling, which is mediated through the Fc epsilon receptor, was thought to be the main trigger, and very important in allergic reactions, for example, in triggering mast cells. But actually, we now know there are lots and lots of different stimuli that can trigger mast cells. And still not much is known about which specific indications those different triggers are important in outside of allergy or classical allergic signaling pathways. So really, there's a lot more to be learned about the mast cells and how many different inflammatory conditions they may be involved in. But, really interestingly, outside of omalizumab, which is the classical, IgE pathway targeting antibody that's been very, very successful, in many allergic disease indications, there are a number of pathways that seem to be IgE independent.
Okay.
So thinking about, how do we target mast cells agnostic of the signals, individual signaling pathways and signaling pathways that may yet to be discovered? Can we trigger the mast cell selectively? And there is a company that's doing it. I mean, there's some very exciting data emerging from the Celldex clinical trials using the anti-c-KIT antibody. And they're, initially looking in things like, chronic urticaria, for example, which is, patients can have spontaneous urticaria where they get, angioedema and hives, and they don't really know what the trigger is. In other patients, it might be induced by cold or by heat, so there are conditions where mast cells are known to be really, heavily involved. But there are some indications that, in things like, atopic dermatitis and in asthma, that actually mast cells may be actually really critically important. The problem is, if you look at the cardinal genes for mast cells, it's c-KIT. So how do we target c-KIT in a way that is not going to target all of the other cell types that express c-KIT, which is critical for survival and proliferation of many different cell types, including things like melanocytes. So c-KIT is heavily expressed in skin cells, olfactory receptors. Importantly, it's expressed through the development of and differentiation of hematopoietic stem cells. So, things targeting KIT can be, really quite dangerous in terms of thinking about neutropenia and thrombocytopenia and taking out multiple white blood cell subsets. So our idea is trying to think about mechanisms that work for controlling mast cells, but doing so in a way that drives specificity to the mast cell and away from other blood cells and other expressing.
I see. And then, so from what I understand from what you're saying, then if you can figure that out, then there's different ways in which you can control those mast cells, right? So you can target different conditions and different situations.
Exactly. And I mean, I think there are many reasons, again, mast cells are incredibly difficult to work with. And I the beauty of the work that has been done published most recently about different subtypes is certainly there are very many context specific gene expression profiles, for example, for mast cells. And, you take them out of the body and it's really, really difficult to recapitulate what might be relevant in vivo. So this idea of pulling apart a system and trying to figure out how to answer one piece of the puzzle has been of critical importance, because there is a very small number, actually, it's a really tiny community that is expert in working with mast cells. So you can imagine finding a CRO that has expertise working with mast cells has been, it's been a puzzle, which is what got me interested in science in the first place, solving problems.
What a fabulous link, Orla, for us to talk about your career. No, that's really interesting. Thank you for that. We've done a whistle-stop tour through your career. You briefly gave us an overview. But actually, so you talked just then about solving puzzles as being where the interest in science came from. Tell us a bit more about that. Why science? Why biochemistry? Why drug discovery, drug development? Why this career for you?
Yeah. So I guess, I mean, I must have been interested in science in high school. I mean, I did, you know, obviously moving into that kind of final cycle, you know, we had to select preferred subjects, and I was doing physics and biology and chemistry and maths. And so obviously there was some sort of interest there, but I was never thinking about the next step, and I never had. And while on paper, I suppose it may look like I had a pretty normal, you know, career development through science, it certainly wasn't that way in my mind. It was always kind of one step at a time. And I think, you know, I've always been very lucky. I think my parents had always just encouraged me to do something that I enjoyed, and that's what I did. And so, you know, I was thinking about what to do, and I was definitely going to university because my brother was there and was having a great time. And I thought definitely this is something I wanted to do. And my first choices were all either English or, you know, science related. And I always preferred the sort of biological sciences a little bit more. And then my brother was ahead of me. It looks like I just copied him. But he was doing natural sciences in Trinity. It's a long time ago now. I mean, this is when you were doing science. It was science. There was very little, you know, it was so broad at the beginning. And actually from being a really, you know, good academically, I think in high school, I went to university to do natural sciences, and I was completely overwhelmed at the breadth of subject matter. And I didn't really do very well at all in my first couple of years. But actually looking back, I think what was great about it, it was so broad. I mean, I think about students making decisions now, and there are courses like, you know, human genetics or structural biology, or how do you know at that stage of your career, you know, exactly what area of science you want to get involved in. So I'm very grateful that it was so broad. It gave me a really good feel for, you know, everything that was out there. And again, I just preferred, I was drawn more towards, you know, biological pathways, I suppose. And I chose biochemistry for specialization. And obviously part of that was getting the opportunity to work in a lab. And I loved being in the lab. And I had a great mentor there. I mean, it was kind of in between my second year of university and selecting biochemistry as my subject. And I got the opportunity to do a summer internship in the lab, where ultimately I ended up doing my PhD. And it was just a great environment. I mean, really, it's kind of, for the first time, there was no hierarchy. You know, my supervisor, we'd all go for coffee, lots of PhD students, postdocs, and I was being exposed to that environment, I think, quite early on. And I just really loved it. You know, he'd just ask us random questions, and there was this real, you know, environment, I suppose, of open discussion and drawing things on napkins and all of that sort of stuff. And I decided definitely I loved being in the lab. I loved research. So that was the only decision I made. I wanted to stay in the lab. And, you know, after my finals, I hadn't even signed up for anything. I didn't have a master plan. There was no particular area of biochemistry I was interested in. I just liked asking questions and designing experiments to try and answer those questions, or at least build up the story. So I mean, it was after results, you know, everybody I'd celebrated, my supervisor sidled up and said, you know, have you thought about a PhD? And I kind of hadn't, but I said, it sounds like a good idea. So I did that, and it was really hardcore, you know, enzyme kinetics, old fashioned, I suppose, biochemistry at the time. And it was tough. I mean, it was really, really tough. It was, you know, PCR hadn't been discovered that long before, and I was trying to clone this novel gene involved in heme degradation pathways, and you know, it was a struggle. And I guess it was a really good learning experience. Most of what I did, and I mean, this may be most of people's experience in science, you know, unless you're very lucky, most of the things I did didn't work. And of course, it's that idea of learning, you know, redesigning, retrying, and gradually, you know, building up knowledge and getting expert in different techniques, and, you know, trying out different things. So again, I mean, I was finishing up my PhD. I kind of knew I did not want to do any more enzyme kinetics. I didn't really know what I wanted to do. And it was a real kind of turning point for me, I suppose, because I realized I had dedicated, you know, 3.5 years to a PhD. I'm a scientist, but I had no idea what I really wanted to do next. You know, and I didn't see myself in a purely academic role. I didn't see myself, you know, in that academic environment where you don't have the luxury, unless you're, you know, in at the top of your game and in a really exciting hot field of science, you don't have the luxury of doing whatever you want to do. I mean, a lot of the time people seem to be struggling for grants and, training students, and you get your students trained up and they're doing fabulously well, and then they leave you, and, you're constantly in this cycle of competitive, grants. And I just thought it seemed like not the environment I really wanted to be in. And again, the split often between teaching responsibilities and research in the lab, that balance just didn't seem to be in the right direction for me. So I was struggling, and bizarrely at the time, the head of our department was doing a lot of exchanges with the University of Siena and the University of Florence, and there was a lot of Italian students coming over, and people from our department had the opportunity to work over in Italy. And I thought, I really like the Italian lifestyle, and I was doing it.
I'm very jealous of that, by the way.
I was doing a night course, an Italian night course. I really love language. And I just thought, I've been in Trinity for my undergrad, for my PhD. It is time for me to leave. And I had the opportunity to do a postdoc in Trinity, but I decided to just go. And, I asked around the department, does anybody have any contacts? I said, I might as well try and stay in science.
Yes.
And I mean, I just seemed to be, just really lucky. Timing was right. One of our PIs in Trinity had a contact with a great cancer researcher over in Milan, and he had just won this European grant, and I had the opportunity to go. So I went, and the contract was supposed to be for a year. And again, it was just really lucky timing. So this was a lab that was based on a hospital, San Raffaele Hospital in Milan. It was a kind of a, cancer cell biology program. We were looking at the urokinase receptor and trying to understand how it mediated, all of these pleiotropic functions. What were the interacting proteins? What were the mutations responsible, or the amino acids responsible for mediating all of these multiple different interactions? It was a really involved project, but at the time the Italian Cancer Research Foundation were really trying to, build this institute which would bring together multiple different functions and core technologies, with the idea of working together to find a cure for cancer. Right. So I really liked that idea, this broad goal, this combined goal of a whole institute. And so they built this wonderful new facility in Milan and brought together all of these core technologies. I mean, it was the first time. It was almost like a small company in a way. They had expert microscopy, they had a sequencing facility, protein production. They had basically what was like a department store. You'd go in the lift with your little basket and take all of your buffers that were pre-made, and the environment for research was fantastic. And they had these huge big open labs, so you didn't share, you were not beside people from your group. You were working with researchers from lots of different core competencies and different skill sets, and from multiple different countries as well, which was a really great adventure. They were really trying to, I suppose, make cancer research within Italy less sort of insular and more global, and they were attracting students from all over Europe. And so it was a great time to be there, and it was a great opportunity to start to understand, I suppose, the strength of a team rather than individual research, because you were getting all of these inputs and being exposed to all of these different technologies and different questions that other scientists were trying to solve from different angles. And so, we took on a really, really broad project where we couldn't have done it. We ended up making, can't remember how many amino acids were in this receptor, but something like 250 different single amino acid mutations. And we made it in the context of multiple different cell lines. And, we did tons of things that as a team, we approached it as a team rather than trying to solve these problems as individual PhD or postdoc scientists. I guess that was my greatest lesson from my Italian experience, which was not 1 year. It ended up being 5 years. I loved it there, and I, kept looking for excuses to stay. So I think that, was a fabulous experience, even outside of the science. I think just, being in a completely different culture, learning to communicate with people from, diverse backgrounds and with diverse languages, and, overcoming some of those communication challenges, but also just simple things like trying to set up a bank account in a different language, in a different country, and simple things that you think, you don't really appreciate the value of them, I suppose, till you're outside of it. But I mean, 5 years is a long time. And again, I think, thinking, well, what next? Am I going to live in Italy forever? Or, you know, and I kind of felt like it was time for me to move on after 5 years. And, you know, it was back before everything was online, and, you know, my dad was cutting out little advertisements from the newspaper and posting over to Italy, trying to get me to come home. And one of them really caught my eye, which was this, you know, drug discovery company, which was established. Because I mean, I knew at that stage, I loved this team environment. I really loved the power of, you know, everything that we could harness as a team rather than independent researchers. And so this opportunity, you know, Wyeth, we're setting up this drug discovery group in Dublin. It was the opportunity to be there right at the beginning. It turns out, you know, I went to Boston for my interview because the group didn't exist. You know, they were setting it up from scratch, and I was ended up being employee number 3. I mean, I got the job. I didn't even apply for, you know, the role that I got was not the job I applied for.
Right.
I ended up being recruited as a kind of head of assay development. You know, and looking at my, you know, my experience to that point, that really wasn't my core skill set. I think, but, you know, it certainly was up for, it sounded like a great opportunity. I got to see the labs in Boston. I mean, they were amazing, you know, the sort of technologies and equipment and automation that was there at their disposal. I just thought it looked like really something completely different, fantastic. And again, really suited me because I wasn't, you know, I wasn't somebody that had a very specific field of research, or all along it had been just about what is available to me to answer this question. And I thought this looked like such a great opportunity because, again, it was kind of a, you know, technology core providing this expertise and this problem solving capability to all of the different therapeutic units, you know, inflammation technology, cardiology, oncology, you know, so really potential to you know, broadly impact multiple different pathways, but in the same way addressing every challenge as a series of questions. So that was a massive experience because really it was going from nothing, from an empty lab, to trying to establish a drug discovery team, and so hiring people and, you know, building the team and training and learning how to use automation for the first time. And yeah, that was a steep learning curve. And of course, the transition from academia to industry was, you know, was something completely new. But I suppose that's when I really realized the years in Italy were valuable in many other ways other than the science, because that communication piece was so important in trying to, you know, get people on our side. I suppose we were this little group that existed in Dublin, you know, why, when the rest of the team were all based over in the US, mostly on the East Coast. And probably everybody wondering, well, what you know, why do we need this team? What are they for? What is their role? And, you know, really trying to win people over, I suppose, and establish the group as a really valuable part of the organization. And, you know, everything was, you know, over polycoms, and, you know, this was before the pandemic where everybody got used to that way of work.
Yeah, of course.
It's our daily life back then. And so, yeah, I mean, that kind of element of communication and being able to, you know, advocate, I suppose, on the behalf of your team. I think that was a really important skill set. So it was super exciting time. I mean, great opportunity, landed on my feet completely, the timing. And I stayed there for a very long time. 14 years in total between
14
Wyeth.
years.
I started in 2006 with Wyeth as a principal scientist, and left. We were acquired by Pfizer in 2009.
Yes.
And I left, you know, combined 14 years later, and went through the ranks, you know, to associate director, manager of the team, and then on to senior director of the group, leading the group in Dublin. I mean, at that time, I suppose when I left, we were probably in the range of maybe 25, 30 people. And we survived, you know, through all of the change, all of the reorgs and restructuring and acquisitions. And so, yeah, it was an adventure.
And I suppose being in that, given the unique context of the group that you were working in, I suppose, within the company, that probably set you up pretty well for life in biotech compared to perhaps if you'd just been based in Boston, or if you'd gone and worked for AstraZeneca in Cambridge, or, you know, been more of a, been closer to the center of the organization. as you said, having to almost justify your existence and show value and figure out what people need from you and how you can contribute.
Yeah.
Works pretty well, I guess, in terms of then thinking about, okay, well, how are we going to go and raise money, and how are we going to engage with patients and all the things that you need to do in a biotech company.
Yeah, I mean, no question. It was a very unique situation because, while we were part of a massive organization, we were kind of operating in a way like a small biotech, because we were so geographically removed from the rest of the team, and we were interacting across all of the different therapeutic units. I mean, there was communication streams in multiple different directions. Plus, we were a small research group that was based at a large manufacturing site. So when we were established initially, we were set up on a university campus, which was a great time. And of course, many of the team, because there was no history of drug discovery and development in Ireland. So there was nobody that was trained, ready to organization. So we were taking people from academia with PhDs and people straight out of their degrees in Ireland and training them. But we were coming from academia ourselves. I mean, it was 3 of us at the time with no industry experience. So the whole thing was really a real learning curve. And we really, I suppose that went into the kind of hiring process. We wanted people, of course, who were smart and intelligent, but a big part of the interview, it didn't matter if they had no relevant experience, because we didn't expect them to. There wasn't any of that sort of environment that existed. But somebody that could come in and that could tell us whatever their scientific story was in a compelling way, we knew that was going to be important in terms of building a team that can advocate. And so then just before we were acquired by Pfizer, I think people in Wyeth felt, well, there's this little team that are on a university campus. They are going to be so easy to just, why do we need them? Let's get rid of them. It's the logistical challenge to have them. So luckily we were moved on to the large Wyeth manufacturing site in Dublin. And while, again, I mean, that was another culture shock and change for us, I think ultimately it was a fantastic opportunity for us, to learn. It's great that we can make these things in the lab, but actually we have to think about how it translates into a 250 litre bioreactor, or a 10,000 litre bioreactor.
Yes.
So that for the first time, I think that was another really big step in our learning is how do we think early on about, is this going to be a drug? Not from the biology perspective, because we've kind of got that bit figured out, but are we going to be able to make it? Are we going to be able to, how productive is it going to be? What are the sequence liabilities that might lead to bioprocessing problems downstream? And so it was a good opportunity, but it also brought a big challenge that the manufacturing site were worried about getting product out. They didn't really care about what probably to them at the time seemed like a group of students just messing around in the lab. So it was weird from the academic side. People thought it's big pharma. We moved on to the manufacturing side. They thought we were university students. We were always kind of in that sort of outsider environment, and that was really critical, I think, in the development of the team on the manufacturing site, was really to be able to tell the story. This is why this group is important. This is what we're doing. This is how we impact. Imagine, and the sort of story we really built on and tried to get people excited about was the idea that something we discovered in our lab in Dublin would one day come back to the site in Grange Castle to be manufactured, and wouldn't that be amazing? And people got super excited about that idea. And so you start to learn how important the story is. So understanding who the audience is, what you need from them, how you can help each other, and then how to communicate that in a way that gets them excited too, I suppose.
Yes. And there may be several ways you can answer this next question, and we've probably touched on one of them, and it might be what you've just said. But through your career, where you've been an academic in Ireland and in Italy, worked in a big pharma company, worked in biotech, and seen lots of different elements of the industry. If you were, I'm sure there's lots of different things that you've learned, but if you were talking to someone who was earlier in their career, who was starting out, is there one thing that stands out as kind of advice that you'd give, or something you've learned that has been particularly important from a career perspective for you?
Yeah, well, I mean, I think communication obviously is a big one. And I think still today, whatever the environment you're in, I mean, thinking about trying to spin an idea to potential investors. I mean, the question is why? Why is this story interesting? Why are you making this drug? Who is it going to help? What's going to be different? So learning really about figuring out what's important, stripping everything back, being able to, everybody talks about the elevator pitch, but I mean, I think that's a really clever way to do it, is to force yourself to tell your story in a really concise way that can be very compelling. But that story has to change for the audience, depending on who that audience is. So understanding that, I think, is critically important. It's the same for everything you do, thinking about sending out CVs, what is the job? Who's going to read this? What are the important things? And instead of giving it out to multiple different life science recruiting teams, you want to really think about who you're sending and how to tailor it for the scenario. So I think communicating in a meaningful way is probably one of the most important things. I think also for me, I mean, I always felt like a little bit, like being a generalist was a real disadvantage. I always felt everybody else knows not just what they want to do, but whose lab they want to do it in. So when I was thinking about what am I going to do next? I've just got my results. I have no idea. I have no plans. I should be much more organized. And other people were thinking about this particular professor's lab they were going to in this particular university in the US or whatever. But I think what's always been important is that, whatever routes you end up taking along the way, there's always massive value in every situation, whether it's scientific. And while my scientific experience in Italy was fabulous and we published some great papers, and we did I learned so much. I think in value to my own personal development, I think, the outside of the science was really the most important thing. So I think whatever opportunity that arises, it may not be on that path that you think you should be on, but it will bring value. And you have to do your best to identify what that value is, I think, so keep an open mind about opportunities and also, feeling like, there's value in applying for something that you think, you may have only 30% of the tick list that they're looking for, because I had pretty much very little for that job, with Wyeth originally, and, I ended up getting it. So I think keeping an open mind and in what an opportunity might bring you, and keeping an open mind when you're, thinking about applying for things, But I think the communication piece for me probably has been the most. Being able to tell a story and understanding who the audience are, I think, is the most important.
I think it's whatever career you're in, I think that's important. And I think sometimes as a scientist, it can be more challenging because you're so involved in the work you're doing, right? You know everything about it, you know all the detail. It's sometimes hard to realize that the audience doesn't.
They don't care. They don't care. You care, and actually, my brother is an academic. He's a neuroscientist. And, we have lots of fun conversations about the merits of our careers, and I think as things have evolved over the years, we can both really appreciate, the value of each other's situations. But very early on, when I was in Italy, and I was so engrossed in this particular project around this receptor and individual amino acid mutations and structurally what it meant, I went to his lab. I had this opportunity. For some reason, the urokinase receptor was of interest in some of the neuroinflammation models he was looking at. So he said, wouldn't it be great to work together and publish a paper together? And so I ended up getting this travel grant and went over to his lab. He was based in Southampton at the time, in Hugh Perry's group. And I was introducing the project to the rest of his team. And for the first time, I was like, oh my God, there's blank faces, no questions. Nobody cared, because they're interested in disease models. They're interested in the whole animal and the response, the biological response, in the whole animal. So they didn't really get it or care. And that was a really valuable lesson for me. I think I'm telling them the wrong story. I should be telling them backwards, this is what's important for you. This is why you should be interested. So that was the first real example of not having a clue, not really researching who my audience was and what would have been of interest to them. So I learned it pretty early on, I would say.
That's good. I think it's a really critical lesson. Orla, this interview has absolutely flown by. Thank you so much for joining us. It sounds like a really exciting time for Granular and for Ultrahuman 8, and we wish you the best of luck with it, and we'll be keeping a close eye.
Thanks, Tom. Pleasure to talk.
Thanks very much for listening. Careers in Discovery is sponsored by Singular, helping you to build a brilliant biotech company. Biotech leaders spend far too much time, money, and energy on hiring and people issues. Head to www.singular-biotech.com to learn how you can recruit and engage your team more effectively so you can focus on developing medicines, treating patients, and saving lives.